everyone. Thank you so much.
Thank you again. I now call upon Dr. Harita Desai to introduce our second speaker for the day.
Thank you, Dr. Vivek. Our second speaker of the day today will be Dr. completed his bachelor's in pharmacy from University of Mumbai in 2004 and was a proud recipient of the Professor M.L. Khurana Memorial Merit Award. He completed his master's and PhD from Institute of Chemical Technology in Pharmaceutics. While working under the guidance of Professor Pradeep Bhavya and in collaboration with University of Turin, Italy, Dr. Shankar pioneered research on cyclodextrin-based nanosponges. To accelerate his research work, he was invited to spend a year in Turin as an exchange research scholar. After his PhD, Dr. Swaminathan worked in the area of combination drug product development and medical devices in Femicare, Mumbai, before moving to University of Tennessee Health Science Center, Memphis, USA, for his postdoctoral research program, focusing on ocular research. He was a very successful postdoctoral trainee, bringing in independent funding for his research ideas in drug delivery to the eye for retinal disorders within 2 years of his appointment. He was later promoted to assistant professor in the same department where he continued his pursuit to make a difference in ocular drug delivery. After about 4.5 years in academia, Dr. Shankar made his foray in industry in US by accepting a senior scientist role in Perigo Pharmaceuticals in Minnesota. He continued working on ophthalmic drug products while also expanding his repertoire in solid oral dosage forms, parenteral and topical systems. He gained experience working on QBD programs and implementing design of experiments in his research while working on complex generic products in Perigo. While serving as SME in aseptic processing and ophthalmic drug products, Dr. Swaminathan quickly rose to the level of principal scientist in Perigo Pharmaceuticals. After spending more than 4 years in Perigo, he moved to a brand pharmaceutical company in Washington, D.C. metro area known as Supernus Pharmaceuticals. In his current role as Assistant Director, Drug Product Development, while reporting to the Executive Director, Dr. Swaminathan oversees new drug development programs in the CNC space. With this introduction, I invite Dr. Shankar Swaminathan, sir, to start the second session. Sir, welcome, sir.
Hi, Harita. Thank you so much for the kind introduction.
Yeah.
Um, I hope I am audible. Um, and I'll start my session. I thank you. So I have, a big task ahead of me, especially after a great talk from Prithipal. And I'll try my best to, introduce this somewhat boring subject, but, I'll try to make it interesting, but because it's very, very important in the industry setup to know what QBD is and to use QBD principles in drug product development. My goal here will be to touch upon the key aspects because there's no way one could complete a full presentation on QBD in 1 hour, but I'll try to touch upon the key aspects while going into details in some of the examples that FDA has provided. And I'll try to highlight what ideas one could apply to academia to make the academic projects industrially relevant. So let's go into my slides. Like I said, my objectives would be to provide a brief background on QBD, and for streamlining purposes, I will focus on the hypothetical example provided by the FDA, which deals with the modified release drug product development. Then I'll also provide an overview of a typical, product development for ANDA, and I'll try to touch upon the 32P2 module, which covers everything that there is to know about formulation development. So when FDA sees a product or an ANDA submission, your 32P2 is like your index, and it's everything that you've done in the product development. Then I'll touch upon QTTP, CQA, risk assessment of drug substance attributes, excipient selection, and I'll also talk about risk assessment of formulation components and a little bit about DOE studies, which is very important, and one could easily apply these principles in academic research. Then I'll go upon and start talking about risk assessment for manufacturing process. However, I will not discuss that in detail in this presentation simply because it's a very lengthy subject and probably deserves at least 3 to 4 hours to go into the details of it. But I will definitely encourage everyone to go into the references that I provide in the slides, to go into the detail, try to spend as much time as possible on those references, which are open source references provided by the FDA. And then try to use those principles in your academic research as well. Then I'll touch upon control strategy, and then in the end, I'd like to wrap up with learnings for academic research projects and what are the potential strategies that I think could help bridge the gap between the industry and academia. Uh, First of all, standard disclaimer is that these are ideas that are presented in the presentation are my own personal opinions and does not in any way reflect any of my current or past employer's position. I am neither funded nor endorsed by my current employer for this webinar. No proprietary information or knowledge is presented in this webinar, and the materials presented are based on published literature and regulatory documents available in public domain. The presentation is solely for educational purposes, and it does not serve as a reference document because there are regulatory guidance documents provided by the FDA. I strongly encourage you to refer to that if you're making a product development decision. So let's jump into the subject, QBD. What is QBD? You might have heard about QBD. These days, thanks to COVID, there are several webinars and workshops conducted, and some of the workshops have really covered QBD in really great detail. So you might have heard about QBD also in academic setting, and also if you're new to industry, of course you would have heard about it. But let's see what QBD is all about. FDA wants when a product is submitted, when a dossier is submitted to the FDA, they want the quality of the product to be built into the product. It does not necessarily mean that if you test more, it does not mean that you have a higher quality product. So they want whatever you do, you think about quality in the end. So but you have your end in your mind when you design a product is what QBD is all about. It's a systematic approach to development that begins with predefined objectives and emphasizes product and process understanding and control based on sound science and quality risk management. Now, don't lose focus, and focus on sound science, because sound science is something that you can never get away from, be it academic research or industrial research, because you base all your observations, base all your development strategy around sound science. What are the goals of QBD? To achieve meaningful product quality specifications that are based on clinical performance. Like I said, you need to have your end in your mind while you develop the product. So you always think about clinical performance and have product quality, which is meaningful. To increase process capability and to reduce product variability and defects by enhancing product and process design, understanding, and control. These are very heavy words. Each of these words carry a lot of weight, and each of these words have a lot of experiments, a lot of thoughts, strategies going into explaining each of these words. We will touch upon some of these in the next few slides. To increase product development and manufacturing efficiencies, and to enhance root cause analysis and post-approval change management. Root cause analysis is a very important subject as well, because several times, in the industry, there are deviations, and deviations occur. Either it can be planned, it can be unplanned, but several times, you grapple with what you're presented with, and if you've done a good job in developing a product, it becomes way easier to identify the root cause analysis of your deviation, and that makes your job very easy, and you can easily rectify the issue and make sure that the product is not compromised from a quality standpoint. These are the basic elements of QBD. It's a quality target product profile that identifies the critical quality attributes of the drug product. So QTTP and CQA, these are two terms that are synonymous with QBD, and it is our basic building blocks of QBD. We'll go into details of each of these terms in the next few slides. Product design and understanding, including the identification of CMA. So CMA is critical material attributes. That is the third pillar of QBD, and process design and understanding, including the identification of CPPs and thorough understanding of scale-up principles, linking CMAs and CPPs to CQA. So, one should always have their focus on the CQA, which is the critical quality attributes of your drug product. and one should never lose focus of the critical quality attributes. But the key is to link your critical quality attributes to your CMAs, which is your critical material attributes, and your CPPs, which is your critical process parameters. So if you are able to link these two things together to CQAs, you've done a good job in your product development strategy. And at any point of time, you can always come back and rectify any challenges, any troubleshooting issues, if you have a systematic design. In the end, a control strategy, which includes specifications for the drug substance, excipients, and drug product, and as well as controls for each step of the manufacturing process. I liken the control strategy to, say, a short, snapshot of your drug product development. So, say if I were to transfer my technology to another company, or say in a different place, this control strategy should be sufficient to give an idea to the new person who's trying to develop this product as to what are the key factors that one needs to take care, well, to make sure that the quality is not compromised and you get reproducible, robust product development. So it's a very important document, and it's a invaluable part of your product development strategy. In the end, process capability and continual improvement. We'll not talk about this in the presentation, given the time constraints. Um, so these are the, this is, you will see this quite often in several QBD presentations and FDA documents. This is, the flow chart of your product development profile. So essentially start with your product development profile, then you identify your CQAs, then provide a risk assessment and do a design space, then, have a control strategy in place and continual improvement. So a lot of jargons, I know it can really get overwhelming at times. So let's, dial back and think about, something we all like to eat, and most of us we like to eat. So let's think about, baking a cake. Formulation development. I liken formulation development to cooking at times, and in this COVID era, you see a lot of posts about cooking and creative cooking and stuff like that. So I think this is a very relevant subject, and let's dial back and forget about the jargons for a moment and think about baking a cake. What goes into baking a cake? And I'll try to highlight the QBD principles from a cake perspective. To bake a cake, you need these materials, like eggs, you need cake flour, you need salt, you need flavor, you need sugar, of course. What are the different material attributes? First of all, let's see what are the different critical quality attributes. No matter how well you make the cake, if it doesn't taste good, it's of no use. So taste and mouthfeel, they are really very important from a cake perspective. Flavor, softness, and sponge-like feel is also very important. You can't give a cake that feels like rock when you put it in your mouth. So you need to have a sponge-like feel. So for me, critical quality attributes, these are critical quality attributes with like taste, mouthfeel, flavor, softness, and sponge-like feel. So what are the material attributes? Like I said, flour is very important. So let's say particle size of flour. Say you have agglomerated flour particles, that may not give you a good cake. Bulk density, water content, flow, particle shape. These are important flour attributes. Then, sugar, of course, is one of the most important materials in the cake. So, granule size, sweetness index of the sugar. Then egg, provides softness and consistency to the cake, and porosity as well. Egg white content and the type of egg used, might be a material attribute. Then flavor, you can use natural, synthetic flavor. Some flavors, leave an aftertaste. You're going to get away from that. And so these, for me, these are the material attributes. And formulation variables, like the ratio of flour to sugar, ratio of egg white to flour, the amount of sugar used, amount of flour used, all these things can affect the way you get your end product, which is your cake. Then the process, of course, is very important, like the mixing speed. You don't want to mix too fast. You would end up with making a lot of froth. You want to use the right type of mixer. Then you want to optimize your mixing time, not too little, not too much. And then the order of addition of ingredients also can have a role to play. And at what temperature you bake and at what temperature you finally take the cake out also is very important. So once you optimize your process using baking trials, and understanding how each of these input variables affect your output CQAs. So by input variables, I mean the material attributes, I mean the critical process parameters, how that affects your CQA. Once you dial down and once you understand how these affects your CQA, you can devise a control strategy, and then you can also devise, CMA and lock down your material attributes as well. Say, for example, there might be a situation where a flour of a particular brand will give you the cake of desired quality. So if I make the cake, it comes out really well. If I transfer the technology to you, you always face challenges, and then we realize, oh, it's the flour that's causing the challenge. So you want to write down what is unique about your flour in your control strategy. So if you get the document and if you get the tech transfer document and you see control strategy, oh, this flour is important, let me make sure that I get the same flour, the same specifications. So that becomes your CMA. Similarly, the process, say, I have a particular type of mixer or a particular type of oven that gives me this particular cake, and kind of relates back to the particular quality of the cake. So you want to make sure that you enter that in your control strategy, so that the other person, using the process, understands this is a critical aspect of your product development. And in the end, your in-process controls, of course, like visual checks and smell check and all that kind of stuff. So you want to make sure that you don't put your cake for too long in the oven and cause a fire hazard. So just to give a perspective of formulation development from a food perspective. So these are some of the aspects that we'll be touching upon from a drug development perspective in the next few slides. Um, so typically, this is a typical progression of a drug product development. So you start with your defining your QTTP, and I would like to touch upon some of these aspects which can easily be translated into academic research as well. So when you start a research project, define what you want to do, define your quality target product profile. So based on your drug substance properties, based on your R&D characterization and the R&D label and the intended patient population, you define your QTTP. Then identify what your CQAs are, like your physical attributes, size, splittability. Now these are from this particular example that I'm going to talk about. Then assay, content, uniformity, and drug release. Then optimize formulation, like excipient selection, feasibility trials. And then once you optimize your formulation, then identify and categorize formulation and process variables as high, medium, and low risks. So risks is very important. Whenever you do something in your life, risk is a very important aspect as well in your general life. In product development, it becomes all the more important because in the end, you talk about your product, which goes eventually into humans. So risk is very important, and you need to have a strategy where you can mitigate risks and have a systematic product development. And so based on your risk assessment, you develop your experimental strategies and experimental data. And in the end, when you have your product which is scalable and which is submitted to the FDA, you want to make sure that you have addressed all your risks and you have your experimental data supporting each of your risk assessments. And then in the end, scale up, where you use your design of experiments to investigate high risk variables and reassess your risk, and control strategy, like we talked about. And this term will come quite often in the presentation as well. So these are the resources I have based my presentation on, and you can find it online. And basically this is the quality by design for ANDAs, which is an example for modified release dosage forms published by the FDA. It's basic resource document that I've used for the presentation. I may have skipped mentioning the source in some of the slides, but I've tried my best to identify the source of my data. If you have any questions, feel free to shoot me an email. I'll be happy to point you to the right resource. And there are a couple of good papers also online, like Understanding Pharmaceutical Quality by Design by the staff of FDA. It's a very good paper. It kind of goes into the details of explaining the principles of QBD. Jumping into QTTP, it's a prospective summary of quality characteristics of the drug product that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of the product. So in the end, all that matters is safety and efficacy of your drug product. Examples, we'll go into the examples in the next slides, but in this slide, I just want to touch upon the intended use in clinical setting, the route of administration, dosage form, and the delivery system, dosage strength, container closure system, therapeutic moiety release or delivery, and attributes affecting the PK properties, and the aerodynamic and dissolution properties of your dosage form. And this has to be the first step of your product development, and that has to be devised early in your development based on the properties of drug substance, characterization of RLD product, and consideration of the RLD label and intended patient population. So, you have your RLD analysis, which is a quintessential part of your product development strategy. You base it upon your clinical pharmacokinetics and drug release information that you already have from the RLD. Now, in this case, we are talking about this particular example. We're talking about a brand modified release dosage form, 10 milligrams, and excuse me, the formulation composed of an immediate release and an extended release component. The label recommended dosage is 10 milligram once daily. So, to explain the QBD principles, we'll be basing our uh, strategy on this particular product. So this is your research product. Consider that this is your research product, and how do you go about developing the product development strategy for your research project. So what do you know about RLD? So we know that it's a 10 milligram tablet. It is an extended release, modified release dosage form. It has two components: an immediate release component and an extended release component. It has a half-life of 5 hours, and it has no food effects. Its metabolite is inactive, and drug release. It has a biphasic drug release pattern, and the IR component, as expected, provides the initial push, initial immediate release, and then the ER component, the extended release component, provides the extended release profile. And the drug characteristics of your whole and split tablets are also similar. Now, analysis of RLD and physicochemical characterization. So, what do you know about RLD? There are a lot of resources, in the ANDA dosage development, in the FDA portal itself, where you can easily find information about RLD, and also, like the, like label information. It kind of becomes a very important part of your generic drug product development strategy. So, some of the information you can find it off of label as well, and the composition information has to be made available. Especially the qualitative composition information has to be made available. So this is your RLD characterization based on the information that is available on hand. So I want to touch upon how you devise drug product development if you don't have an RLD. Say, for example, you're starting a research project and you don't know an RLD. Excuse me. So in that case, it is highly recommended that you use publications. Excuse me. Available publication information, and available research information that's available in public domain as your starting point if you want to start your drug product development in your academic research. So in industry, if you have an RLD which is already available, then it's very easy to find information about that. But QTTP, analysis of RLD forms a very important part of your QTTP, since you'll come back to this at all the time, all the point of time in your drug product strategy. So RLD composition, what the RLD is composed of. This is somewhat reverse engineering, and also basing somewhat on existing literature. So for this particular product, it's a hypothetical example. This is what the RLD composition looks like. It has ER coated beads, and it has the IR granules and the final blend. So the ER coated beads, as expected, has a drug, has a binder, which is PVP, has ethyl cellulose as your coating polymer, and polyethylene glycol as plasticizer. It has a sugar sphere, which is a substrate, and the IR granules has MCC as your filler, and lactose as a filler as well, and then sodium starch glycolate as a super disinfectant, and magnesium stearate as lubricant. It's a very standard formulation, and it's a hypothetical example. One important thing I want to touch upon is the importance of using IIG, which is your inactive ingredient database that FDA publishes every quarterly. They update this, or there is a frequency at which they update. And I highly recommend even academic research professionals to use this database when they devise their academic projects, simply because if you make a research idea into a, say, industrially scalable or translatable product, you want to make sure that you lie within the IIG levels that the FDA has prescribed. So you don't have to rework your formulation. Say if a particular industry is interested in your product, you want to make sure that your levels are within the IIG levels. Then going into the details of QTTP, how do you define a QTTP? And I would highly recommend devising your QTTP for your current research project. So after this presentation is over, kind of reflect upon what I talked about, go back to your resource materials that I mentioned, and think about your research project, because you yourself are the best judge of your research project. You know more about your research project than anybody else. So basically devise what you think your quality target product profile could be. Now in this particular case, dosage form is modified release dosage form, and in a generic drug product development, it's very important that it has to be a pharmaceutical equivalent requirement. Route of administration, so in this case it's an oral formulation, and dosage strength 10 milligram. Pharmacokinetics, in some cases in academic research you may not have pharmacokinetics readily available, but in this case we have the pharmacokinetics available, and we know that the pharmacokinetics requirement, it has to be bioequivalent. And stability, and this is a very standard stability definition of 24-month, and drug product quality attributes, and this is very important, and we will be using this, and this kind of is carried forward in your product development strategy as you move forward, like the physical attributes: identification, assay, content uniformity, degradation products, residual solvents, drug release, microbial limits, water content, and then the container closure system. It's very important from an industrial standpoint. I would highly recommend also think about container closure system academic research as well, especially if you have plans to collaborate with an industry partner, and eventually make a foray into translating the technology into clinic. Also administrative administration, and concurrence with labeling, especially in generic drug product development, label has to match with the RLD, and the alternative methods of administration, in this case it doesn't apply. Then physical attributes like the appearance, odor, size, score, and splittability, especially in tablets, extended release tablets, which has a break line in the formulation. Then friability, has to be less than 1%. Identification, it's a positive for drug substance. You want to make sure your formulation in the end is containing the drug substance, because in the industry there are several products being made at the same time. Although there are strict checks and balances, there's always a possibility of cross contamination and major errors. So you make sure that it's positive for identification. Then assay, content uniformity, and degradation products. So as you see, you may have observed, while I was discussing the QTTP, I also had CQA table here, a CQA column, so that one could see whether it's a CQA or not. So if you see here, physical attributes. Like appearance, although it's important, it's not a CQA, because it doesn't really matter if if your appearance is different, but in the end, if it gives the safety efficacy, which is which is what the most important thing is. Size, in this case, definitely matters, because tablet size correlates to swallowability, and therefore it's critical for comparable ease of swallowing, as well as patient acceptance and compliance with treatment regimen. So size is important, and we will talk about this as we move forward. So the idea is to kind of go back into the QTTP and kind of tease apart these drug product quality attributes, and and ask yourself if this is a CQA. And so beginning with size, yes, size is a CQA. Score and splittability, yes, it's a CQA, because the RLD tablet is core and labeled for half dosing. You may have seen, in your label as well, if you have, in your home, if somebody's taking a sustained release formulation, it will say do not crush, or do not half the tablet, or it will say that if you want to break the tablet, use the break line. So it's important, and it affects your safety efficacy. And there is a reason why there is a break line, in a formulation. Identification, although it's important, like I said, it is not something that we will discuss in detail, because there are checks and balances in your drug development strategy that will take care of your identification. Then assay, it's very important, and we will talk about assay, and it's a CQA essentially. Content uniformity is a CQA, because to have a formulation which gives reproducible safety efficacy, and is robust, you need to make sure that your CQAs are taken care of while you develop your product. Degradation products, it's very important, but we will not touch upon this in this particular example in greater detail. And then drug release is one of the most important aspects of any drug release, any drug product development. And in this case, it's equally important. So we will be delving into greater details of drug product development. and and we will talk about whole tablets, split tablets, and alcohol-induced dose dumping. In this particular example that FDA has provided, dose dumping is very important, and we will touch upon this. And it's a CQA for this particular example. Microbial limits is important, but you will have checks and balances in place, and justification is one of the other important aspect, which I'll highly recommend use in academic research as well. Because say if you're defending your thesis, or if you are trying to answer queries of a reviewer who's reviewing the publication. If you have justification, designed as you are developing your drug product, it becomes way easier in the end. Say, if you submit a paper and it takes a while before you publish, it gets eventually published. So it may happen that you already moved on, and you are busy with your jobs, and it might be hard for you to come back and look into what you designed, and what ideas went into developing the dosage form. So it's highly recommended to have justifications in place and document these justifications while you're devising your academic projects as well, so that it helps you as a reference document essentially. So risk assessment, low, medium, and high. These are three risks that FDA has defined. A lowest is a broadly acceptable risk. It's a green light. You want to see more of this green light in your product development strategy. No further investigation is needed. Medium is highlighted by yellow. Risk, there is a risk. It's acceptable, and if you do further investigation, it's fine. But if you don't do, that's also fine. But the red ones, they are the ones that might come and haunt you, while you're making your product. So you want to make sure that you address the red ones, with utmost detail and as much as data as possible. So how do you go about, doing your QTTP, or how do you go about using principles of QBD? It's a very systematic approach. You start with your drug substance. Drug substance is the most important part of your tablet, and without the drug substance, your product is useless, virtually useless. So start with your drug substance and focus on your CQAs. So the CQAs, uh, the CQAs in this case, as we discussed, are the physical attributes like the size and splittability, and assay, content uniformity, and drug release. And focus on the drug substance attributes. The drug substance attributes, like we talked about, so in drug substance, think about all attributes that you can possibly think about your drug substance. An example tip here is use the CFA of the manufacturer, to use your drug substance attributes, and also look into literature to see what important drug substance attributes can have an impact on your drug product CQA, because this is very important. Not lose sight of your drug product CQA, because your drug product CQA is the most important. And so these are from this particular example: solid stage form, particle size, bulk density, hygroscopicity, solubility, residual solvents, process impurities, and chemical stability. These are the important drug substance attributes, and how that correlates to your drug product CQAs. So in terms of solid state form, the impact of solid state form on the physical attributes, that size and splittability, is low. Impact on assay is low. Impact on content uniformity is low. But when you talk about drug release, it's high. And we will talk about justifications as to why it's high, because a very important part of your QBD is rationale and justification. And also in terms of solubility, it's very high when the impact of solubility on drug release. Um, Justification is kind of goes hand in hand with your risk assessment tables. So let's see solid state form. Physical attributes, assay, and content uniformity. The solid state form does not affect the tablet properties, which is size, splittability, assay, or content uniformity. But solid state form does affect drug release. And why does it affect drug release? So in this example, my goal is to kind of inculcate thought process as to how you can use what the FDA has given in your academic projects. So what thought process went into saying that the solid state form has an high impact on the drug release? So let's read this. It says the drug substance, and this particular IR tablet contains the crystalline drug substance Z, and no solid state form transformation occurs during manufacture, which is good. And however, the drug substance may convert into its amorphous state during drug layering process used to manufacture. And example, the MR tablet. And this transformation may impact your drug release. Say, in this particular example, drug layering is done wherein you have your drug which is coated onto a substrate. In your academic example, it may be making nanoparticles, it may be solid dispersions, it may be nanosuspensions or whatever. So, your formulation may have an impact in the eventual drug release. What you do, the iterations you have on your formulation may have an impact on your final drug release. So it might be the case where your solid state form of your API might have a high impact on your drug release as well. Then, moving on to particle size, bulk density, the impact of that is pretty low for all the CQAs. Then hygroscopicity, again, it's again pretty low. What I also recommend is while you start thinking about these aspects in your formulation, uh, you know, you have to repeat this process several times, because in the first instance, when you start working on a project, everything, excuse me, everything might come up as a high impact on your final CQAs. But as you learn more about your product, as you know that people have done this before, there is literature available, you may feel better, and you may say, okay, the impact is not that high, but I might probably make it medium. Or then, if yeah, I mean, it's a slam dunk, people have done this for ages, let's go and change it to low. So I want you to think in that perspective when you devise your academic projects as well. Moving on to the further justifications, like the residual solvents, process impurities, chemical stability, all these are low, and I won't go into the details in the interest of time. So the next step, which is very important, is your excipient selection. And one thing which when I was in academic, field, one thing I did, and I was, I think, highly recommended to do, is do an excipient compatibility. It's a very simple, straightforward experiment, but it saves so much effort. In the end, say in your stability, 6 months down the line, you have your thesis submission deadline, and then you have a surprise in your analytical data. You see uh, a wicked RLD, sorry, a wicked impurity peak coming up, and then you're kind of back to square one. You can struggle, and then you go back to your advisor, ask for more time. It kind of creates a lot of problems for you in the end. So if you do your homework properly and do your excipient compatibility, you will get a lot of ideas about compatibility of your final formulation. If you run excipient compatibility, it can be done very easily, 1:1 and 1:10. So pretty standard combination, standard ratios. So mix your API with your excipient, or proposed excipients that you want to use, subject to different conditions, and take samples out of different time points, check for your assay, check for your degradation products. If you want to if you want to be smart, and if you want to go the extra mile, you can also do a DSC, TGA, FTIR characterization of that. And these things also help in publications of data as well. And especially it helps if you want to translate your technology to industry. And in this example, there are IR and ER components, and these are two examples. I won't go into details in the interest of time, but you get the feel. You have to make sure that your compatibility is established before you finalize your excipients. Formulation development, this forms the bedrock of your formulation development. So identify your drug substance and excipient attributes. Like, going back to the example of cake, you know what you want, your end product, what goes into making the end product. So why not, tease apart everything that goes into your formulation, and understand how each impacts your CQA, and use risk assessments and do a systematic development and in the end develop a control strategy. So in this particular example, they have a very systematic approach, and we will go into the slides and let's understand the process flow to begin with. So in this particular example, there are two components, an IR granules and the ER coated beads. So the IR granules, uses a very simple technology of high shear granulation, and the IR granules are formed, and then you add your cushioning excipients, and then you compress tablets, and then of course you have your ER coated beads where you do a drug layering and then polymer coating, and then you blend and you compress. It's a very simple process. But then when you look at the formulation components, this is how a risk assessment table looks like when you talk about formulation components. So like we talked about, the main parts are the IR granules, the ER layered pellets, the ER beads, which are the coated pellets. So you do drug layering, then you do coating with the functional polymer, and then you add extra granular excipients. As you see, there are a lot of red color here, which is not bad. Which is, I highly recommend having a lot of red high risk parameters in the beginning of your product development, so you can address each of the aspects. But the most important thing is, it has to be a very systematic approach. So assay is very important, and the ER beads. If you do drug layering, the layered beads, it has a high impact. If you don't layer it properly, if the drug is low, it will have an impact on your assay. It will have an impact on your drug release. It will have an impact on your dose dumping. Also, similarly in ER coated beads, these are high factors. So I will probably skip a few things here, in the interest of time, but I'll touch upon the important aspects. I'll make sure that we don't miss anything. So when you talk about IR granules, it's an important step, but there are not many parameters in IR. We'll focus on the ER for this example. And the assay, as we said, it's very high. Like I said, the binder type and the level, as well as the viscosity of the drug layering solution, will impact the adhesion of the drug to the beads. So if you have a highly viscous medium and you're trying to coat the drug onto your substrate, and it may not coat properly, so your assay will be low. So it has a high impact. So you want to make sure when you develop your formulation, you address that aspect. Similarly, the drug release. If you have, say, two viscous, or if you have a higher amount of, one of your components, then your drug release will be impacted. So you want to address that as well. Similarly, for the coating of the ER beads with a functional polymer, the polymer characteristics are very important, and you want to make sure you address that as well. Also, the extragranular excipients in this case have some impacts, especially in the content uniformity and the drug release. The type of extragranular lubricant and disintegrant will eventually determine how the tablet is disintegrated, and as a result, it can change the rapid release of the IR portion, and it can also have integrity of your polymer film during compression. So you want to make sure, especially in this case, you have your coated beads, and you want to make sure that the in compression the coating is not disrupted, so that you get the same release profile that you hope to get, which is extended. So these things have to be addressed when you devise your dosage form. And as you read this example, try to think and reflect upon what you're doing in your current projects, and try to use examples. It may not be as extensive at this, because in your research you're trying to do something very disruptive and novel. But still, I would highly advise, if you start thinking about things, you would be very, very surprised to know that you know so much about your product. And, in this way, you will even get ideas to kind of go forward with your research. And then initial risk assessment of your ER polymer coating is very important. Like, this table forms a bedrock of your next steps, which is your drug product CQA. You take that, and then you look at the drug product intermediate CQAs, and then formulation and process variables. So in this case, that there is a drug layer on top of the MCC bead, and then there is a rate controlling polymer. So initial risk assessment of your ER polymer coating. Now we are going into the details of all these reds. This particular column here, the ER beads, this particular column, the ER beads coated, and then the extragranular. So next step is to delve into each of these reds and try to see if we can change it to yellow or better green by supplementing with experimental data. So talking about ER polymer coating, the critical polymer coating level is very important. The polymer aging is important. The additional pore formal level is important. The plasticizer level is important, and it has a significant impact on your drug release and drug release alcohol dose dumping. See what we did there. Now we have only 2 CQAs. You might ask, why not 4 CQAs? This is because in this particular process we identify your intermediate CQAs, and hence instead of having all these 4 drug product CQAs, now we are left with 2 drug product CQAs. That's what systematic development does, and which is very important. And then then you start talking about justification as to why the polymer coating level is important, and providing sound justification is very important. And then also looking at the literature that you have in understanding how you decided to choose a particular polymer. In this case, the hypothetical example uses hydrogens and the Collicoat SR30D, and elasticity, the TG, the glass transition temperature, and the mechanical strength and mechanical properties of the polymers, and the film forming temperature. And these things are very, very important in a polymer coating. And these are the factors you consider while devising your polymer formulation. And the elongation, the break, and all these physical attributes, which you may have learned in your physical pharmacy, it definitely is very important, to use what you learn in your graduate school and undergraduate school in your research programs. And you will be very surprised at some of the principles from, say, physical pharmacy, Martin and Lackman. You can easily translate in your research, and kind of form a novel idea and to characterize your formulations. And Also the coating polymer. One of the most important factors which some of us neglect is, depending on your vendor, your excipient manufacturer. And I think Samir talked about excipients yesterday. I kind of peeped into it. Very productive session. So excipient vendor, they are very important part of your product development as well. You can rely upon their data and rely upon the documentation. So use that as much as you can in your product development as well. I'll skip these slides, but I'll jump to the DOE studies, which is very important. And many times people think DOE is QBD, which is not. As you know, QBD is now a systematic development, thinking about QTTP, CQAs, and all the risk assessments. But DOE is a very important part of QBD. So it's very complicated if you look from the outside. But when I was starting up, I thought UAE, oh my God, I don't want to venture into that. But when I started doing it, it was so easy, and I thought, why didn't I think about this before? And I really encourage all of you to think about it when you devise your research projects as well. So first, you need to understand what are the key factors or input variables in your DOE. That is very important. So in this case, we're going to talk about the pore former, the level of plasticizer, and the extent of polymer aging. And you need to know what you're going to base it off of. So in this case, it is the output, that is the drug release, T20, T50, and T80. So Minitab and GIMP, they are the two invaluable tools in a DOE study. Some of them are also available as a free demo version. I highly recommend you to use those, and there may be others available too. Not recommending these. This is only from my personal experiences. And when you feed in the values, you have -1 and +1. So -1 is the low level, and the +1 is the high level. And then you have your A, B, C, D, which are your factor formulation variables, and responses are your Y1, Y2, and Y3. So when you feed in, this is what the software spits out. It says these are the runs. You do these runs, and in the end, if you do these runs and if you subject your formulations and find your drug release, you will essentially know which is significant, which is not, and what concentrations you want to use. So it's very important to do DOE studies. Like these graphs can be sometimes be very overwhelming, but when you look into it in greater detail, they're not as overwhelming as it looks like. So A, B, C, these are the three factors which have come up as significant in this particular DOE study. So when I say significant, what I learned is, the farther they are from this particular line, the more significant it is. So in this case, they are really far, A, B, C, and then they are labeled also. And if the software labels it, decides to label it, it becomes significant. Um, and also, this is very important. It kind of gives your design space as to how you can work, and design space from a formulation standpoint. So your levels of your TEC and the levels of your theoretical polymer coating level. Anything that is green is what you like. You don't want to go into the red area. So in a layman terms, try to stick with green. Then once you do your DOE, now you feel comfortable. Okay, I've done my homework. Let's see if I can do a risk assessment again. And this process is repeated again and again, in your drug process development. So when now the drug release is medium, because now you have some assurance that, okay, now I have some standards put in, like the polymer concentration, the level of my TEC, and now I am pretty assured that now I can dial down my risk level to medium, and better in this case, like my risk level is low in some cases now. And then again, going back to the risk assessments, so if you go into the FDA example, they have really gone to the details of each of these aspects. In the interest of time, this definitely deserves more time. I don't want to do injustice to this topic by quickly skimming over things. So I won't go into the details of each of these aspects, but I highly recommend you. It's a 161-page document that FDA has, and I highly recommend you to study that and translate it into your product. Maybe a nanoparticle you're working on, it may be a transdermal patch, but there are ideas which you can easily translate. And feel free to shoot me an email if you have any questions. I'll be happy to help out in terms of translating these ideas into your research project. Manufacturing process development, it's another important aspect of your formulation. And just like your manufacturing, just like your formulation, you need to identify each of your possible material attributes and how that affects your process. And you will hear this term a lot, which is the critical process parameters. And the critical process parameters, they form the bedrock of your manufacturing strategy. I'm just going to go over the high-level slide that the FDA has presented in this example of what a manufacturing process would look like, simply because I want to kind of impress upon you the thought process that went into devising this. So you go back to your original CQAs, which is your drug product CQA from your QTTP table, and then you kind of match that with your process. So when like going back to the example of cake, you have your mixing. So in this case, it's the IR granulation, it's the ER drug layering, it's the sieving, it's the beads, polymer coating, and then the blending and lubrication and compression. Going back to the cake example, you have your mixing, you have your baking, the way you pour into the tray, then your oven parameters. So those are the critical parameters, the time at which the baking is done. So in this particular example, the drug layering, and kind of match each of your CQA with and relate each of your CQA to your process steps. In this case, they have found several high variables, especially in the compression side, which is a very complex process. Although we learned about it very early in our pharmacy school, but there are several aspects to compression which will impact, especially in this case when you're trying to compress an ER granule, where the compression forces can damage your drug layer, and it can change the way the drug is released. So there are a lot of high variables in this case, and in the example that you will see, they have done systematic experimentation to dial down this high to low and medium. Like I said, I won't go into the details of this. I really want to quickly touch upon control strategy. We've talked about this. I won't go into the details of how a control strategy is devised, but this is single document that kind of gives you a snapshot of what you did in your product development strategy. It's highly important, and I highly recommend you to have this control strategy in your thesis, in your academic research as well. Although it might sound very boring and non-innovative, because you want to do innovative things, use state-of-the-art technologies to characterize your formulation, which is very good, very important, no doubt about that. Nothing can compromise that. But having something like this will definitely add value to your thesis as well. It kind of gives you've thought about everything. When you see a control strategy document, and when I started my industry career, I was so impressed with this control strategy that I thought this is the single most important document that you should go into the details when you're kind of researching your R&D, or researching your product, because this will tell you everything that you need to know when you want to make a formulation. Like going into the examples, say coating dispersion, holding time. Now you devise your holding time. FDA wants really, they really want a lot of data. So holding time in this case, they had an 8-hour holding time, and they have a range of 0 to 48 hours. So in your control strategy, what you do is you write down in your batch record what is required. Say a particular condition that you want to maintain. The operator wants to know that. You yourself will not be there on the shop floor when the product is commercialized. As a researcher, you depend a lot on collaborators and different people from the different departments. Especially the operators, they are highly trained, but they may not be as scientifically trained as you. So you want to make sure that you dial down everything in your control strategy, so that the line leads and the managers kind of translate this to your operators, and they know what they are looking for, and you have all the information in your batch records as well. This is what a control strategy looks like. Now, in the last 5 minutes, I'll quickly touch upon what we can do to bridge the gap between industry and academia. And I made sure that I touched upon some of the aspects while I was discussing the slides. So know your goals very early in your research work, and draft product development report as you go, and using the principles of QBD. Also, I highly encourage you to talk to industry colleagues and never hesitate to get their thoughts. Also engage industry personnel as co-advisors and external consultants, and have a CDA in place if you don't feel comfortable. Pritipal kind of talked about that, in terms of transparency. Sometimes, having a CDA kind of, is very reassuring, so that, you can freely talk to your industry partners. Collaborate with excipient manufacturers. Like I said, it's a very important part. The information that they can provide to your research project is going to be very, very important. Sometimes they may not take academic researchers that seriously, because in the end, they are very busy and they work with a lot of industry clients. But if you tell them, the importance of your project, if you tell them that, how my project can be translated, and if you can pitch, accordingly, they will definitely be interested. Use DOE wherever possible. It's add enormous value to research publications. Also use good documentation practices. Needless to say, very, very important. Whatever you do, make sure you use GLP, use audit trails wherever possible. Calibrate your lab equipment. Sometimes in research labs, it's like, typical chalta hai attitude, which may not work in your research these days, especially with, scientific misconduct and importance of, your GLP, GMP. Make sure you calibrate your, equipments, and use daily checks, and use robust and appropriate controls, which is very important. And also use appropriately powered statistical tools. As Ronald Fisher said, to consult a statistician after the experiment is finished is often merely to ask him to conduct a post-mortem examination. So never use statistics after you've done your experiment. Use statistics as you're doing your experiment, or even before you start your experiments. And also, especially for new professors and assistant professors who are starting their research career and starting the lab, and who have startup funds or grants, you set up a lab with a healthy mix of innovative and disruptive projects, and include industrial transferable projects as well. And this is true from my own personal example. I worked in City ICT with Professor Bhavia, and our lab had a healthy mix of people who could think out of the box, and people who could bring in their industry experience, and people who could use their industry experience and teach other people in the lab how to use industry principles while devising research projects. So I highly recommend new researchers to use this strategy. It's a very, very fruitful strategy. And use all these principles we talked about while devising your project. Regularly meet with industry personnel. It may be an informal coffee or just a phone call. Some of your colleagues may have industry jobs. Talk to them. Sometimes it helps to kind of open up your thinking process, and also keep abreast of your FDA resources. FDA has been really good in the past decade or so to update their resources, and there are a lot of free resources, even training modules available online, which you can easily read in your free time. And check existing commercial product landscape. This is something I didn't do that much when I was in my academic research, which I regret now. But I highly recommend you to look at what the existing pending PDUFAs are. PDUFA is the new drug approval process. So if you look at that, and you can base your research project based on your pending PDUFAs. So industry might be interested because they are looking for next big thing. Especially generic companies, they want to make sure they are kind of in line with what is coming up. So if you base your research projects based on existing PDUFAs, para 4 filings, and ongoing clinical trials, like use clinicaltrial.gov document website, and see what clinical trials are ongoing, and base your research project. Especially if somebody's starting a research project, these tools are invaluable. And also look at a lot of investment banks and their investment theses. There are a lot of good ideas there, and one could look at that. And there are no substitutes for publications and patents, although some might say that publications are not that relevant in industry, but it is relevant to build your reputation. So if you want to work with industry, collaborate as a consultant, you need to make sure that you have a stellar publication record in good publication, good journals, because that's all that will speak for you. And characterize formulations extensively. There is no substitute for characterization. Provide adequate resources to lab personnel. This might seem very silly, like you're setting up a lab, and why am I talking about resources? Simply because if you want your lab personnel to follow GLP, to follow documentation practices, you need to make sure you provide them with supplies. You need to provide them with printer. You need to provide them with hard drives. It may be a moot point in established countries and established lab, but I know still know that there are places in the world where there are not supplies. There is no internet in some places, to be honest. But then make sure that you provide supplies, hard drives, and make sure that you kind of facilitate all that is required to make sure that GMP is followed. Attend workshops as much as you can. Devise a lab website, which is very important these days, to kind of get your name out there. If you want to collaborate with industry, get your name out there. Do not hesitate to do that. Some of us are introverts. As a researcher, some of us are introverts, but I'll highly recommend. There is no shame in publicizing your own research. Be factful, be truthful. Don't be a braggart, but at the same time, make sure that you touch upon what you do best. Volunteer to organize events like such as these events, which are very important. Like now I know several people in academia, in the current academic field, and me, and we can always keep in touch. So these kinds of events are very important. Funding matters. It's very important to your research, but at the same time, sometimes you have to do some things for free. Sometimes an industry partner might approach you, say that we don't have funding at this point, but can you quickly test this? Sometimes you may say, I'm busy, I don't have the time, but sometimes it might be worthwhile to do it. Sometimes I'll go back to Pritipal started with a mythological example. I'll go back to Bhagavad Gita as well. And sometimes you have to do your things and don't expect anything in return. So these kinds of things might help you in the future. And I have seen from personal examples, like in my PhD days, I used to work on industry projects. I never thought how those projects would help me in my thesis, because they didn't really have any direct implication. My thesis subject was completely different. But I can tell you from personal examples, the stuff that I worked on really, really helped me in the future. And even what I'm doing today, they are very, very important for me now. In your grant applications, try to include industrially relevant, equipments, like funding for those equipments, so that you build a lab with industrially relevant equipment, so that you can do industrially relevant work. Now I know there are several students, undergrad students, and what you can do best to kind of chart your career as an industry professional, or as an academician, or a academic faculty, to have collaboration. First of all, get good grades. Unfortunately, that's still the milestone that one looks at. Get good grades. There's no substitute for that. Read your books properly, understand it, stick to your passion, follow your passion, attend industry relevant workshops. Although it might be cliche, but passion is very important. And volunteer to do industrial projects in the lab. So as an undergrad student, there might be a faculty member in your department who's doing breakthrough work in working with industry. Tell them, I would be very interested in shouldering some of your research fellows, and I would like to learn. It definitely does a world of good if you are pursuing, if you want to come to US or abroad, for further studies. It will also help you in bolstering your CV if you write a publication with them. like one of your academic faculty members. And so these things are very important. Also, use the free training modules on FDA portals. These are very very important tools. I wish I would have known about this before, but now I know, and I still do. I still learn every day. As I'm in the industry, I still consider myself as a student, and I make myself abreast with all that is available in the portal. So in the end, I want to thank everybody, especially Clara and the organizing committee for this webinar, and Bombay College of Pharmacy. There are fond memories with Bombay College of Pharmacy, going back to undergraduate days and the faculty members and the RX. I'm very, very honored to be here and make this presentation possible. And whether you like it or not, we have to thank COVID for this. Because mankind, if you are presented with a challenge, you come up with solutions. So as Pratipal mentioned, this online portal wouldn't have been possible. One wouldn't have thought about streaming if it weren't for this COVID era. So I thank the organizers of BCP for inviting me, and IPA for inviting me as well. Thank you very much, and I'll be happy to answer your questions. You can feel free to send me an email, connect on LinkedIn. I'll answer questions as and when possible. Thank you.
Thank you so much, sir, for the very lucid talk on QBD and how it can be applied to academic projects. So, you started the session with helping us understand the basic definition of QBD, followed by various QBD principles, and you actually explained us very easily with the help of a cake what are the jargons involved in QBD. And I'm sure we as academicians can take this up in imparting this knowledge to the students. You've then elaborated on how generic product development progresses, right from analysis of RLD further to planning of CQAs, understanding QTPP, and you have very rightly impressed upon us the need for excipient compatibility that is actually a source of many problems even in academic projects. Finally, you have supplied us a nice example of a DOE study with some interpretation which has helped us gain more confidence in now applying QBD to our research projects. And your last slides about actual QBD application to research projects as well as your advice to students has been really helpful to all of us. In the interest of time, sir, I'll just take up two questions. The first one is by Pradnya V, and she's asking there are various risk assessment methods mentioned in literature. Is there any selection criteria for it? And what is the name of RA method that FDA recommends in its guidelines?
That's a very good question. When I was starting up, there were a lot of information available online, like, fishbone structure. There is a lot of Japanese principles of, Six Sigma. There are a lot of systematic approaches, and feel free to use those. The more you use, the better it is to kind of, come up with, QTTP and risk assessment tools. But from a personal example, I like to rely a lot upon prior data and prior example and prior knowledge. Some of the knowledge, it's available online. You just have to look. So I really base a lot on the prior knowledge available. But feel free to use systematic approaches like Fishbone, wherein each of the parameters is hashed upon, and you understand each of those parameters and in the impact on your CQA. I liken risk assessments to a brainstorming session. Say, I would also encourage you guys to do that. Say, have your project team in a room, have a board, practically write each of your CQA and QTTPs, and ask people. Different people have different ideas, and all of those ideas are very very important. Like when I started out, it was highly encouraged even from the FDA as well to have a group discussion on these parameters. So include all the stakeholders. In an industry, you have different departments like QA, RA, the tech transfer, the operations. So involve everybody early up in the process. Take their views, take their thoughts, and devise your formulation. So from the other aspect of RA tool, from what I understand, it probably the question the person is asking what is recommended by the FDA. I don't think FDA has recommended any
Yes.
particular tool, but they do
Okay.
say that feel free to use it as long as it's systematic, as long as it uses sound science. I don't think there should be a problem. Thank you.
Okay. In the interest of time, I'll just take one last question. I request the audience to directly post your questions to Sir. Sir has provided his email ID. So it would not be possible for us to address all the queries on this platform right now, but request you to ask or get in touch with Sir directly. So the next and the last one is by Harsha. She asks how detailed should reverse engineering of RLD for generic product development be, and what analytical tools usually are used for its characterization.
That's another wonderful question. Uh, so let's take an example. Since we've already talked about solid dosage forms, let's take an example of a topical dosage form, say an ophthalmic ointment for that matter. So if you want to talk about RLD characterization, you can get a lot of information from literature, like the label itself, which is available on on several websites and also an FDA portal. They are the brand manufacturers are supposed to mention the key ingredients in the label itself, so what it contains. So most of the times it might contain a petrolatum, a mineral oil, maybe a preservative. So those are the three main ingredients in a topical dosage forms, especially in ophthalmic, oleaginous or hydrophobic ointment. So you know
Yes.
that, but then you don't know the grade of it. So sometimes information like
Right.
that can be easily obtained from some people might have done literature. So literature search is of prime importance. But at the same time, tools like
Okay.
MS, LC, MS, those tools can be used to understand your profile of your hydrocarbon base, profile of the ointment, the base that has been used. Then you can also
Right.
use NMR tools and several characterization tools to understand the excipient characterization. But in terms of the API, you can always try to form a process wherein you can extract the API out of your formulation and make sure it's not affected. So like a non-destructive tools, there are several tools available to do a non-destructive characterization of the API. So from that you can understand the polymorphic form of the API. You can understand the crystal behavior. You can also understand the particle size potentially. Sometimes it is practically impossible to do that, but you got to live with what you have and
Yeah.
try to do as try to go into as much as details as possible. So there is no real right answer to what
Yeah.
details you can provide, but use your best judgment, use sound science, and have use whatever is available online, and also do high throughput experimentation and characterization to characterize your RLD.
Yes, I think this one can be real challenging, and you've really provided us great inputs on this. I hope this has answered the question. I now formally close this session. Thank you so much, sir, for having spent your time with us and all your expertise and your opinions. They've really provided us great knowledge today in terms of QBD from industry point of view and how we can apply them to our projects in academia. So again, thank you so
Thank
much,
you.
sir, for having here with us on behalf of audience as well as the organizing committee and Bombay College of Pharmacy.
Thank you very much. I'm honored. Thank you.
We've come to the end of our session. Our webinar is towards finishing. I just want to request all the participants that the feedback link is posted in the description box under the YouTube video itself. However, it will be activated only at 6 p.m. So please be patient and wait till then. Once it is activated, we will let you know on the WhatsApp group. I now call upon Dr. Clara Fernandez, the organizing secretary, to formally close the function and deliver the vote of thanks. Over to you, Clara.
Thank you, Dr. Vivek. Good evening, everyone. It is my privilege to propose a vote of thanks on behalf of Department of Pharmaceutics, IPA-MSB's Bombay College of Pharmacy Autonomous. The organization of any event is generally an outcome of concerted efforts of every individual. It is my great pleasure to express, on behalf of the organizers, a deep appreciation for the timely support and encouragement provided by such individuals. At IPMSB's Bombay College of Pharmacy Autonomous, we are highly appreciative of the leadership and the extensive support provided by our management, especially by Mr. Nitin Manyar, Chairman of IPMSB, and Dr. N. Shiva Prasad, President of IPMSB. We place on record our gratitude to the incredible leadership and the freedom in decision making provided to us by our in charge principal, Professor Krishnapriya Mohan Raj. Encouraged by your positive feedback, we can confidently say that the success of this 3-day webinar on bridging the gap between academic research and industry commercialized outcomes is primarily because of our distinguished speakers. We are indebted to the speakers for sharing their acumens on the theme by covering various aspects of technology transfer. It is safe to say that the insightful talk has been instrumental in redefining these trying times to transforming times. Indeed, as outlined by our speakers, a plethora of opportunities awaits us. It's only we who have to take a step towards making a difference to our career. On this note, we are immensely grateful to all our participants for your enthusiastic presence on all 3 days. We understand the changing norms of the working hours in COVID 19 times, yet your presence has been a constant source of motivation, and we look forward to conduct another engaging discourse for enabling our unified growth in knowledge and skills. Last but not the least, special thanks to our convener, Dr. Ujjwala Shinde, HOD of Department of Pharmaceutics, for spearheading the organization of this webinar. We also thank our organizing committee comprising of spirited members Dr. Vivek, Dr. Harita, Ms. Shubhangi, and especially Dr. Sagar for working relentlessly in bringing the content digitally to the participants. We are also grateful to we are also grateful to Professor Nagasenkar and Professor Mala Menon for being our mentors and providing constant guidance. With this, I conclude the vote of thanks, wishing each one of you a safe and prosperous journey in bridging the gap between your research and the commercial outcomes. Have a great evening. Take care.